what I'm going to talk about next is a paper that was a collaborative project between Choi in Korea and my laboratory. We published in early 2020. And the title of this paper is The Detection of the Small Oligonucleotide Products of Nucleotide Excision Pair in UV-irradiated Human Skin. So the focus of this is really to look at actually measuring a specific output of the nucleotide excision pair system in the context of human skin. So the basic idea of how nucleotide excision pair works is that we have damage that's induced, for example, by UV radiation. So one of the most common forms of DNA damage we experience is UV radiation from the sun. That induces these thymine dimers in DNA. And the only way human cells can remove that is through the process of nucleotide excision repair, where enzymes come in, recognize the damage, and then cut out the damage. So there's enzymes that actually make what we call incisions, where they cut the damaged strand of DNA, this top strand here, and essentially release that damage in the form of a small DNA, about 30 nucleotides in size. That leaves a gap in the DNA that can be filled in by using the corresponding template of DNA synthesis and then ligation to fill in that gap that gets generated. And so one of my interests over the last few years has been to look at the fate of these small excised DNAs. In a couple of papers, I've referred to them as SED DNAs for small excised damage-containing DNA. They're also called excised oligomers and various other names in the literature. And again, what we've been interested in over the last few years is to measure these and detect them. Other labs have sequenced them to map DNA repair throughout the genome. And so the purpose of this paper is to determine whether or not we can actually look at these and detect these in the context of a human tissue. Thus far, most of the work that's been done has primarily been in the context of cell culture, which has been very valuable. It's been extrapolated or been also studied in mouse tissues in response to cisplatin, but nothing so far looking at in the context of human skin. And so what Juna and I was to set out to develop this assay to look at that in the context of skin. And the basic idea here was to use discarded surgical skin, primarily from abdominoplasties, which has been a nice source for skin for these sorts of studies. We simply expose the skin to UV radiation. We'll take a small punch biopsy. And for this work, I'll show it's about a 6 millimeter punch biopsy. So if you look at the end of a pencil, that's about 6 millimeters. So it's a very small amount of tissue we're working with. We do a heat shock to separate the epidermis from the dermal layer of the skin. So we're specifically looking at the epidermal layer, since that's what gets damaged by UV radiation. Once we have the epidermis, we can lyse the tissue open, and we're going to isolate soluble DNA. So the bulk of the genome DNA is in very big, large molecules, and that can be pretty readily separated from other cell components by centrifugation at high speed. So once we get soluble DNAs, we need to enrich for the damage-containing DNAs. What we've done here is to look at the 6,4 products. So UV radiation induces cyclobutane pyrimidine dimers, or CPDs, and 6,4 products. 6,4 products are maybe 20% or so, or depending on the wavelength of radiation that gets induced by UV. CPDs are much more abundant, but they take a little bit longer to be repaired. They're not as recognized as quickly as CPDs. And so for the work here, we're going to focus simply on the 6,4 products since they are repaired more rapidly than CPDs. Once we immunoprecipitate them, separate them from any other DNA in the tissue or from the tissue lysate, we will end label them with biotin. So biotin is a small molecule, which we can then use HRP streptavidin to detect. And so this biotinylation occurs. We use an enzyme called terminal transferase, which can add nucleotides to the 3' end of the DNA. So what we do is simply add a biotinylated nucleotide to the ends of these small DNAs. So again, we're looking at these small DNAs at 3' end. We can then add a biotinylated nucleotide. And then what we can do with this is to then separate them from any other higher molecular weight DNAs in the cell by running on a gel. So PAGE stands for polyacrylamide gel electrophoresis. We separate things by size. We then transfer the DNA from a polyacrylamide gel onto a membrane. And then we use HRP streptavidin. So this will then bind to that biotinylated nucleotide. And then we can detect the signals with a chemiluminescent substrate that gives off light that we can then detect on, for example, a ChemiDoc biorad system. And this is an example of this data, what we see. What we add to all the labeling reactions, we will all usually add a standard DNA that just ensures that the labeling works well and we don't have anything else in the reaction that inhibits the enzymatic labeling by terminal transferase. So we add a 50-mer single-stranded DNA to these reactions. So we see that in both the samples from the non-irradiated and the irradiated samples. But what we only see in the UV irradiated skin tissue are these said DNAs. These DNAs that range in size from about 20 to 30 nucleotides, which is consistent with everything else we've done in cell culture. So it looks like what we see in human skin looks very similar to what we would see in cell culture, which is expected, but nice to actually show for the first time. The nice thing about this assay is, again, it's a very nice signals to noise ratio. In the absence of UV, there are no said DNAs, no excised oligos being generated in the skin. So again, that was after a single UV exposure, 800 joules per square meter UVB. I believe it's 1 hour after UV. And this is predicted to be a good time to look at the 6,4 photoproducts since they get repaired quickly. 800 joules per square meter UVB is a little bit more than maybe 2 minimal erythemal doses for somebody with my type of skin. We could generate a minimal erythemal dose refers to sunburn. So this type of UV would presumably give me a little bit of reddening in my skin the next day or so. So with that assay, working, we wanted to be sure that this is actually dependent on the nucleotide excision repair system. If we were doing this in cell culture, what we would do is use a cell line from a patient with xeroderma pigmentosum. So there's a number of people with mutations in genes involved in this nucleotide excision repair system. And these fibroblasts are readily available. And you can then test various experiments, various ideas comparing the effect of UV, the effective repair on whatever it is you're looking at. In cell culture, we've certainly done those sorts of assays. What we did here with skin is a little bit trickier. It'd be a lot harder to get skin samples from patients with xeroderma pigmentosum. So one way, in a paper I published in 2019, was we could essentially mimic that with a drug called spironolactone. So this is actually an FDA-approved drug. It's used for a couple of different clinical purposes. But what we found is if we take the skin biopsies and then maintain them in a little bit of cell culture medium for a few days, we can add spironolactone to that medium. The spironolactone goes into gets taken up by the tissue. And spironolactone was shown in 2014 actually to cause a rapid proteolytic degradation of a protein known as XBB. So XBB is required for nucleotide excision repair. So if you don't have it, you don't have nucleotide excision repair, you won't generate these excised oligos. And that's been shown in cell culture. So here what we want to do is to look at this in skin. So we treated skin samples ex vivo with spironolactone, and then did a Western blot of the epidermal tissue. And we can see that very nicely these XBB levels are significantly reduced. There's some variability from subject to subject, potentially maybe due to the background levels of XBB that are expressed. But we can definitely reduce the level of XBB that's expressed in the epidermal tissue by treating with spironolactone. Again, consistent with the idea that XBB is required for nucleotide excision repair, if we treat the skin with spironolactone, it significantly reduces the amount of said DNAs, the excised oligos that are generated after UV exposure. And again, there's some variability from subject to subject, but largely, it's only 4 samples here, but it may correlate with the extent of depletion of XBB is the prediction there. So again, it looks like these excised oligos are generated. We can detect them readily in the skin, and they're dependent on a nucleotide excision repair system. We next wanted to look at sort of the dose response. And so what we did here was to expose a skin to various doses of UVB. Again, non-irradiated, 0 UV. 200 joules per square meter UVB is again maybe a little bit less, maybe half an minimal erythemal dose of UV. And then we go increasing amounts. And as you would expect, it's largely dose dependent. There is some variability. Part of this is sort of the processing of these samples. There's a number of steps, as we showed in the first figure, that potentially you could lead to loss of material from one sample to the next. They may lead to some of this variability. But what we did is to essentially do this in multiple sets of samples to get a better representation of the average. And you see a very nice dose-dependent increase in the said DNAs that are generated after UV exposure. And it seems to saturate maybe 1,600 joules per square meter UVB, more or less. So there may be a maximal amount of repair that can be generated or that can take place in the skin within an hour after UV exposure. And we next wanted to look at the kinetics of this response. And so what we did was to expose skin to 800 joules per square meter UVB, then harvest cells at different time points from 15 minutes all the way to 12 hours. And these sorts of experiments have been done in cell culture, and they show very similar patterns. The degradation of the oligos that we'll talk about in a minute, maybe it's a little bit slower than we expect, than we see in cell culture. But if we look here again in non-irradiated skin, there are no said DNAs. Within 15 minutes, we can definitely see these said DNAs being generated. So repair is taking place. It seems to peak about an hour after UVB. That's consistent with what we've seen in cell culture. Then at later time points, you start to see that this, what we call the primary product, that's closer to about 30 nucleotides in size. Sequencing and other analyses suggested the peak was really about 26 to 27 nucleotides in length, in terms of the length of the excised oligos. Again, at later time points, that seems to be decreased, probably due to all the 6,4 protoproducts have been essentially removed from the genome. They're in these said DNAs. And then what we see is the secondary product form. And these are potentially partially degraded. So they make maybe trimmed either the 5' or 3' into a smaller product. Based on work that's been done in vitro and in cell-free systems and also in cell culture, the primary products seem to be largely bound to the TF2H enzyme, which is XVB is actually a component of that. And TF2H is involved in unwinding the DNA to allow the nucleases to come in and cut. However, the secondary product is only based on the data we've generated, it seems to be bound to a protein known as RPA, or replication protein A. RPA is a single-stranded DNA binding protein. We really don't understand this transition from the primary to secondary product and how that happens, but it seems to be consistent in cell-free systems and in cell culture. So that's what these two populations of said DNAs are, what they represent in the skin. It again seems to mimic pretty well what we see in vitro, both cell-free systems and in cell culture. And in figure 4B and C, what we've done is to essentially quantify the kinetics of the primary product versus secondary. Consistent with what we see in cell culture, again, it seems to peak half an hour to an hour after UVB exposure. Then the primary products start to get degraded. The secondary products seem to peak a little bit later, 4 to 6 hours later, and then they do start to get degraded. And we haven't done a whole lot of work to try to figure out, understand how these oligos or how they get degraded, what the nucleases are that do that. That's still an important question, because there potentially could be defects in the degradation process that may contribute to some human diseases, for example. But again, this degradation seems, based on if you look at data from cell culture, it seems to be a little bit slower than what happens in cell culture. And we don't really know why that is. Again, this is an ex vivo system, so there might be other things missing. But it is human skin, so that's one of the advantages of this assay, is we're actually looking at these repair products in the context of a human tissue. And this was the first to do that, and that's, again, one of I think the strengths of this paper was just to finally show that we can actually detect these oligos in the skin. And one of the ongoing projects are to potentially sequence these and look at sort of mapping repair genome-wide.